Based on a research, the central premise of the Jurassic Park films, that mosquitoes can preserve and provide access to animal DNA, has been validated in a contemporary, practical context by scientists from the University of Florida. While extracting viable dinosaur DNA from fossilized amber remains firmly in the realm of science fiction, new studies demonstrate that modern mosquitoes act as effective, flying samplers of ecological biodiversity. Led by entomologists Lawrence Reeves and Hannah Atzma, a research team conducted an extensive field study in a protected Florida reserve over 8 months. They captured over 50,000 mosquitoes from 21 different species. By analyzing the blood meals from several thousand female mosquitoes, the only sex that consumes blood, the researchers were able to identify the DNA of 86 different vertebrate animals. This represented approximately 80% of the vertebrate species known to be in the mosquito's diet range. The detected animals spanned a remarkable ecological diversity, including frogs, birds, and large mammals like cows, encompassing native, invasive, endangered, arboreal, tree-dwelling, amphibious, and migratory species. The primary strength of this method is its ability to provide a broad, efficient ecological snapshot. Traditional biodiversity surveys are often costly, labor-intensive, and require significant taxonomic expertise. In contrast, mosquito sampling can passively capture data on a wide range of species simultaneously. The study found that during peak mosquito activity seasons, this indirect DNA sampling method was as effective as direct researcher surveys in detecting animal presence. However, the research, led by biologist Sebastian Botero Cagnola, also noted that traditional methods retained an advantage during dry seasons when mosquito populations are lower. Not all species were detected. Notably absent were the endangered Florida panther and smaller subterranean animals like the eastern mole. These gaps highlight that the method's effectiveness is influenced by mosquito feeding preferences and the accessibility of host animals. Nevertheless, the technique proves exceptionally sensitive to a broad spectrum of species, whereas many conventional detection methods, like camera traps or auditory surveys, are often tailored to specific animal groups. The practical application of this research is significant for conservation in what the authors term a critical era of biodiversity loss. By analyzing environmental DNA, eDNA, from mosquito blood meals, scientists and wildlife managers could develop a cost-effective, innovative monitoring tool. This is particularly valuable in remote, dense, or difficult-to-survey habitats where mosquitoes are abundant. It offers a way to track species populations, detect the presence of invasive or endangered species, and inform ecosystem management decisions with minimal direct intrusion. Reeves acknowledges the widespread disdain for mosquitoes as pests and disease vectors, but argues that this research underscores their underappreciated ecological role. Beyond their function as pollinators and food sources, mosquitoes can serve as vital bioindicators. Their feeding habits inadvertently create a dynamic library of the local vertebrate community. In conclusion, while this mosquito DNA sampling technique requires further verification across different global ecosystems and climates, it holds substantial promise. It transforms a notorious insect into an asset for conservation biology, providing a novel method to monitor wildlife and potentially help prevent contemporary species from suffering the same fate as the extinct dinosaurs famously envisioned in the films that inspired this line of inquiry. Both supporting studies were published in the journal Scientific Reports.